So last time we spoke about Shabab, let's start. Last time we spoke about tumor suppressor genes. This lecture will be talking about oncogenes. And let's start. We say there are 100 trillion cells in the body, 100 trillion cells in the body, and all these cells, they proliferate without issues because we have very good control on the DNA damage. And we spoke about the P53 as the guardian of DNA. And we told you that when there is any DNA damage, the P53 will check that before the cell go into synthesis and proliferation. So there are 100 trillion cells. What I want to tell you that these proto-oncogene and tumor suppressor genes, they are normally present in the balance. Proto-oncogene, tumor suppressor genes are present normally in balance, and they don't allow any overgrowth or undergrowth cells. When there's mutation of these, then problem arises. So, oncogenes are mutated forms of cellular proto-oncogenes. We say that before. Proto-oncogenes code for cellular protein which regulate normal cell growth and differentiation.
Yes.
This is the proto-oncogene, the normal proto-oncogene, how it functions. So I want you to understand that very well, because based on that you can understand the oncogene. You have growth factors. The growth factors come to the receptor. They attach to the receptor, then through the cytoplasmic signal transduction protein, they go to nucleus and they stimulate the cell growth genes. Yes, which are the oncogenes. The proto-oncogenes. We are speaking about normal proto-oncogenes. So you have the growth factors, the growth factor receptors, the signal transduction protein, and you have the nuclear proteins, and then you have the oncogene. So all this in normal, the proto-oncogene, how it will act. Of course, there's balance between it and the tumor suppressor genes. So if there is a mutation in any of these, cell they don't act independently. Cell they act, they proliferate, they go into the cycle as a response of the stimulation from outside, from other cells. That's why we have growth factors. The cell communicate between each other by growth factors. The cell communicate inside the cell by the signal transduction proteins. So cell-to-cell communication by growth factor, cytokines and other things. But inside the cell, we have the signal transduction pattern. But the cell, they won't move. The cell it won't go into division unless it's asked to do that. And that's the difference between this cell and tumor cell. The tumor cell is autonomous. They don't need to be asked to go into cell cycle. Whereas the normal cells, they have to be asked to go to cell cycle by the growth factor. And so you have to understand this concept. The cancer cell, it acts independently, autonomous. The normal cell, no, it won't act. No cell will go into cell cycle without the growth factor stimulation. So there's good communication between cells. If you take, listen to me, if you take normal cell, and put it into a petri dish to proliferate, it will die. It will proliferate. Whereas if you take cancer cell, it will proliferate. Autonomous. Nutrition. So what happened in oncogene? There are mutations in either in the genes that will cause secretion of growth factor. So growth factors are always secreted, abnormal. Or the genes that have abnormal receptors. So the receptor is always active. That's what happens in oncogene. Or in the cytoplasmic signal transduction system. So that's always active. So that's what will happen. So there are these oncogenes that produce proto-oncogenes that produce these proteins, because remember we are not talking about the genes. The genes won't do their function. Somebody asked me about the genes and proteins they were producing. The genes are the information, the data. The one that do the work is the proteins. You know the molecular biology dogma: gene, messenger RNA, protein. So this is the central dogma of molecular biology. So the genes are just information. They won't do any work. The gene has only the information. The one that will do the whole work, enzyme, growth factor, all these things are the proteins. And so when we are speaking about mutation in genes, we are speaking about oncoproteins, abnormal proteins. واضح? So these are the four. So we spoke about P53 as the guardian of gene, and you know why? Because it's protect the DNA, and if there's DNA damage, it will stop, arrest the cell, or push the cell into apoptosis. The commonest mutation in oncogene is the KRAS. KRAS mutation. This RAS mutation. The RAS has the family of RAS 3, K, H, and N. NRAS, KRAS, HRAS. But I will RAS family, RAS. So they say this is one of the commonest. 15-20% of cancers have KRAS mutation or RAS mutation, and it's in GI, pancreatic tumor. 90% of pancreatic cancer, GI tumors, and lung cancer have RAS mutation. And in this case, you have the growth factor receptor again. And the inactive RAS with the GDB become active with the GTB. Once it's mutated, it becomes all the time active. And this is signal mutation. And you have the activation of MAP kinase pathway. This is an important slide that I want to understand. How proto-oncogene become oncogene. So I'll spend some time on this. How proto-oncogene become oncogene. Three main mechanisms. Translocation, amplification, point mutation. How proto-oncogene become oncogene. And the translocation, okay, who tell me about translocation? Translocation. Well, anybody, you don't have to come here, just translocation. Translocation. نعم واحد جالس يعني عندك two chromosomes, one part of DNA from one chromosome to go to another chromosome. So you focus معي شباب. You have two chromosomes. Part of DNA from one chromosome goes to the other. Yes, simple time. So because part of DNA goes to the other, the other part, the gene, it gets extra chromosome, it becomes always active. This is active translocation. Typical example of this, I'll give you examples of each one. Then you have to remember these examples. Then we have gene amplification. What is that? Number of copies. We have 2 copies from each gene. In gene amplification, we have 20, 30, 50 copies of each gene. Gene amplification. Multiple copies, not only 3, 4, 10, 20, 30 copies of gene instead of 2 copies. Point mutation. Yeah. Point mutation is, so there's 3 point mutations. Substitution, one deletion to the other, deletion. So, again, substitution, deletion, and under substitution we have 3 types of mutation. Silent.
Yes.
What's silent mutation? The same protein. We don't care about the mutation if the protein is the same. Because the protein is the working force. So the silent mutation means the protein is not changed. Then what other types?
Sense and nonsense.
Yeah, missense and nonsense. What is missense?
Missense is will affect the
Will not
protein.
Yeah.
No, so we have three types of substitution. Silent, no change in protein. Missense, there is what? For example, they use sickle cell. But sickle cell is not tumor, so we'll talk about tumors. And the third one is the nonsense, where it's substitution. So this is all under substitution. Then the other ones were insertion and deletion. There will be preemption.
Yes.
Preemption. Yeah. So there will be preemption, deletion, and substitution and insertion. So these are the permutations. So these are the three mechanisms where proto-oncogene become oncogene.
But then, doctor, when does amplification occur?
I will show you examples of the tumors that have amplification. So these are the mechanisms. Any question on mechanism? Shabab, any question? Mafhuma? These are the mechanism, proto-oncogene, the normal oncogene become oncogene. Yeah. Anybody has? So the three mechanisms, then I'll give you example that you have to remember. These are the techniques that we detect amplification and translocation. One of them, you don't have to remember, this fish technique, fluorescent in situ hybridization. Does Tuha fish? The fish is the fluorescent in situ hybridization. We can detect translocation and amplification. And that's how it looks. By fluorescence. This is the... So, now one of the shabab, now I'll give you example of the different techniques, the translocation. One of the first oncogene to detect translocation is Philadelphia chromosome. Chronic myeloid Philadelphia chromosome. And the gene that's involved is the BCR gene, and it's 922. This I want you to remember. In chronic myeloid leukemia, you should remember 922 translocation, and the gene name is BCR gene. Philadelphia, and the chromosome is called, the results Philadelphia chromosome. From 9 to chromosome. The other one I want to remember is Burkitt's lymphoma, and involve a chromosome 8 to 14, because always there are two chromosomes, the DNA from one chromosome to another chromosome. So it goes from 8 to 14. And the gene involved is CMYC gene. And the third one is follicular lymphoma, because these are the common that we have everyday practice. We practice them in the lab. 14, 18 translocation, and the BCL. Okay, BCL2. Can I have BCL2? What BCL2 does?
One of the families.
One of the families where?
The mitochondria.
Yeah, which, yes. And the what? And the apoptosis.
B53.
Yeah?
It inhibits the formation of the pore.
It's apoptosis, so it's inhibit apoptosis, it's pro or against? Anti-apoptotic?
No, it's pro. It causes people to use
Yeah, because it's common sense. It's pro, pro or anti should be anti-apoptotic because in cancer. No, BAX is that causes apoptosis.
Yeah, that's the talk of that.
No, BCL2.
The anti-apoptotic.
Anti-apoptotic, because we have cancer. And in cancer, we have all the cancer anti-apoptotic. The BAX will cause apoptosis in intrinsic bacteria. So again, we have one of the mechanisms, the proto-oncogene become oncogene is translocation. We have three examples, and this example we selected. These examples are CML, CML leukemia, we have the chromosome, and 922 translocation, and the oncogene is BCR. BCR, but P is double P. Burkitt's lymphoma, the oncogene CMYC, 814 translocation, and follicular lymphoma, 1418 translocation, and the oncogene is BCL2, one of the apoptosis. Yeah.
Yes.
The three examples only. Three examples. So all what you have so far is the three. Shabab,